Hello everyone, welcome to this tutorial on BLAST algorithm. This presentation is designed as a step-by-step guide to undergrad students. I am Dr. D. Anand and today we will explore BLAST, what is BLAST, how it works and its various applications in bioinformatics. Let's begin. So BLAST stands for Basic Local Alignment Search Tool. It is a widely used bioinformatics algorithm that helps to compare biological sequences, including DNA, RNA, proteins. The primary goal of BLAST is to find regions similar between the sequences, making it useful for tasks such as sequence alignment, gene identification, and evolutionary studies. Researchers use BLAST to compare unknown sequences with the existing databases and to identify potential matches. Let's move on to types of BLAST. BLAST comes in different types, each designed for specific types of sequence comparison. BLAST N compares nucleotide sequence against nucleotide database. BLAST P is used for protein sequence against protein database. BLAST X translates nucleotide sequence into protein and searches against protein database. T-BLAST N searches a protein sequence against a translated nucleotide database. T-BLAST X compares translated nucleotide sequence with each other. Each of these variations helps in different biological investigation, from identifying genes to studying protein functions. So, how the BLAST works? BLAST follows 5 steps process to find best sequence matches. Algorithmically, the BLAST breaks the query sequence into k-mers, that is short word fragments, and then it finds matches in the database using what is called as a scoring matrix. There are different kinds of scoring matrices. Most popular ones are PAM and BLOSSOM. Further, it extends the alignment by linking the matching regions, and then it will calculates the statistical significance using E-value to determine the readability of the match. Post this, there will be displaying of results based on alignment score and significance. This process ensures that BLAST provides a rapid and efficient sequence comparison. Let's quickly look into the steps for using the BLAST online. Open NCBI BLAST. You can open it from https blast.ncbi.nlm.nis.gov and choose the appropriate BLAST tool. For if you are using nucleotide sequence, then go for BLAST N. If it is protein sequence, then go for BLAST P. And enter or upload your sequence. You can, if you have your own sequence, you can upload it to the window, or you can search for a sequence that you want to study and then submit it there. Then next step is to choose an appropriate database like NR or NR database. NR stands for non-redundant, or RefSeq database, reference sequence database. Or there are multiple genomes as reference database that you can use. You can adjust the parameter if necessary for better sensitivity, and then click BLAST and wait for the result. So this online tool is widely used in genomics and molecular biology research to get deep insights about the sequence. It's a quick small primer before getting into the video, where I will explain give you a demo of how the BLAST is performed. Open BLAST N. I am going to use Open BLAST N on NCBI. Enter sample DNA sequence. Choose NR non-redundant database and then click BLAST. And we will be observing top hits and interpret the alignment scores. Now let me play a small video, I've recorded a video for performing BLAST. Let's see that, and I'll give you a commentary on how it is performed. So initially we will search for NCBI. In NCBI we will search for a nucleotide sequence. In this case I am going to search for alpha amylase, this choice of my sequence from rat. So you have got several hits of this sequence. We'll choose one of the sequence and wait for the entry to open. So this is an NCBI entry with unique accession number from which we will retrieve the sequence. I'll go click on FASTA at the top and then open the sequence alone. Post this, I will select a fragment of sequence or entire length. This meanwhile, I will also open the blast page, blast end page for doing the exercise. Now blast is open. I'll select a small fragment of sequence that needs to be performed blast. So here I'm selecting a small fragment of nucleotide sequence from of alpha amylase from rat, and I'm pasting at this region where your sequence is pasted. Post this, you can, if you want to say trim off or only select a region which you want to analyze, you can do it from to here, represent this. I'm just giving a job title as strip 2 and then selecting non-redundant database as choice of my database. I'm going to look for highly similar sequences and then now I press blast. Let's wait for the result. So this is how your results would look like. You will have you will have a page like this opening up where description, graphic summary, alignment and taxonomy of the sequence that has been blasted given, and below that you will have sequence with significant alignments will be given at the bottom. Let's wait for it to load. Yeah, so you can see at the bottom all the significant hits that have been generated are present here. Now you have different parameters like maximum score, total score, query coverage, e-value, percent identity and accession length. So we will be seeing what is this one by one to get the interpret the result. So you can see. So this is your alignment page where you can see your query getting aligned to the entire database. Now let's quickly run on to the points that we will be covering on analyzing this result from the previous demonstration that we saw. So first, query coverage, that is percentage of query sequence aligned, and e-value, that is probability of obtaining similar alignment by chance. Lower the e-value, better. It means 0. If 0 is the e-value, that means there is only one occurrence of that particular sequence in the entire database. Score represents the alignment quality. Higher the score, better the alignment. Identity, that is percentage of identical matches between the query and the database sequence. The graphical summary helps us to understand what is the percentage of identity or how much similar are these sequences. So here you can see I've taken a screenshot of the result page to explain you. So you can see the first hit is the same hit that we took for us doing the blast, and the max score is 3027. That is equivalent to the total score, and it's also query coverage is 100%. That is 3027 regions have been matched and they have got a score of 3027. E-value 0 represents there is only one entry of this particular match. So minimal the e-value, better your alignment. Then the percentage identity is 100%. So you can see there are also other entries where the percentage identity kind of varies and other parameters also vary. So this way you can choose the best aligned region for your sequence that you have used for blasting. So coming to application of blast, so this helps to identify unknown sequences comparing genetic sequences across species, finding homologous genes in different organisms, analyzing mutations and evolutionary relationships, and majorly assisting in medical and forensic study. To summarize the entire video, BLAST is a powerful tool for sequence comparison. Different BLAST types serve different purposes. Key results include E-value, identity, and query coverage. And BLAST is widely used in genomics, medicines, and research. Thank you for patient listening.